Welcome everybody, and thank you very much for joining us today on our virtual event. My name is Dr. Alan Mullen. I'm a product specialist for microscopy cameras at Andor, and I'm going to be your host for this live presentation. Today we're going to be talking about experimental bioaccumulation of coronavirus in bivalve molluscs. So I'm delighted to welcome today's speaker, Dr. Giuseppe Aprea. Dr. Aprea is an official veterinary officer at the Experimental Zooprophylactic Institute of Abruzzo and Molise G. Caporale, IZSAM, it's just easier to pronounce, based in Teramo, Italy. The group at IZSAM acts as a public health institute for the Abruzzo and Molise regions, performing analytic work for the public veterinary services and also supporting veterinary and public health across those regions. Giuseppe gained his PhD at the University of Naples Frederico II on the topic of lytic bacteriophages and their impact on the production of buffalo mozzarella. As part of this, Giuseppe also spent time at CAFRI at the Lochry campus in Northern Ireland, as well as at the microbiology department at the University College of Cork. This allowed Giuseppe to develop a broad range of techniques and a broad perspective on the study of microorganisms. After his PhD, Giuseppe then went on to continue his experiences across microbiology, veterinary and food industry areas, and this has proved very beneficial in his current role at IZSAM as an official veterinary officer. In this role, Giuseppe has been involved in the study of numerous foodborne pathogens such as norovirus, hepatitis E, and bacteria such as Campylobacter and Listeria monocytogenes. And looking specifically at how pathogens such as these, as well as pesticides and microplastics, which people are more familiar with these days, may enter the food chain. To do this research, Giuseppe has been specializing in molecular biology and other phylogenetic techniques for the detection and the characterization of the pathogens we've mentioned. Most recently, Giuseppe and the group at IZSAM have been involved in studying aspects of the recent SARS CoV-2 pandemic. The group, for example, has conducted studies on the occurrence of coronavirus and bivalve molluscs experimentally, which we're going to talk about shortly, and also in the surveillance of SARS CoV-2 variants within the region to study the persistence and infection loads using these molecular biology techniques. Just a little housekeeping before we get started. You've all joined this presentation in listen-only mode, so if you have any questions during the presentation, please just type them into the question box in the bottom left control panel as we go along, and then we can get those questions to Giuseppe then at the end. So without any further ado, I'm happy to hand over to Dr. Aprea.
Okay, so thank you to Dr. Mullen for this very kind presentation, and thanks to everybody for your attention for the moment. So I'm very delighted to be here with you today and to bring you some of my experiences in research also for SARS CoV-2 apart from my general topics of interest. So as Dr. Mullen said, I come from IZSAM, which is a body. I just want to give you an overview of how in Italy the Ministry of Health works and what are these institutions from which I come from. So IZSAM in particular is founded in 1941 and is part of the National Public Health Service, and we support the Italian Ministry of Health. It is important to know that I am a vet, and all the expertise of a vet, which are in Italy centered on food safety, animal health and welfare, control and prevention of zoonosis, and support of food industry, all these things are behind the control of the Ministry of Health. And the IZSAM, like you can see in this picture, is located in Teramo, is a headquarter of a big institute working for two Italian regions, which is Abruzzo and Molise. Well, in Italy, we have 10 big IZS, 10 big headquarters, which are the colors, larger colors you can see. And each headquarter is connected to a network of small other labs. So in total, in Italy, we have more than 100 labs from the public authority that we cover all the provinces in all the regions, all the Italian regions. And the field of activities in general of these big institutes are covering the animal health and welfare, the food safety, health and environment, more later also plants, and epidemiology and risk assessment in general. In particular, our institute also manages information systems like the animal identification and registration system. And then we also support the knowledge management. The main task of this research institute is above all focusing on research on etiology and pathogenesis of animal infectious diseases and zoonosis, of course. We know that 75% of the human diseases are coming from animals, so are zoonosis. We also assess the development of innovative diagnostic methods, so we try to be updated for using the most sensitive and specific diagnostic methods, sometimes also producing by ourselves. We sometimes, for such disease, produce vaccines, and we also have microbiological and chemical tests for the disease of animal and in the framework of the control of safety. Other main tasks are the surveillance and risk assessment in animal health and food safety, the surveillance of disease. We also cover an important aspect, which is the training of professionals in the health system. And we support the process of accreditation and certification of laboratories, because as you may know, our analyses are performed under the accreditation umbrella, so we are accredited for our tests. And we do also research in animal welfare. Each headquarter can own some national reference laboratory or international reference laboratory. In particular, in Teramo, we own the Listeria monocytogenes, the Campylobacter National Reference Laboratory, the Dioxin and Polychlorophenyl's National Reference Laboratory, for animal disease, and we also have a reference center for veterinary epidemiology, information and risk analysis, National Reference Laboratory for brucellosis, and for urban hygiene and non-epidemic emergencies. Also for the exotic diseases like the African horse sickness, the Capripox viruses, the Petit ruminant, we own national reference laboratory. So we are a bit the Italian lab which faces the potential coming of new foodborne or animal-borne disease from other countries, but from Africa. Very recently, we also enlarged another lab, which is the whole genome sequencing lab for the microbial pathogens. And we are also enlarging our national services for the databases of animal identification and registration systems. So in particular, we register all the animals that are breeded in Italy for food consumption. Moreover, we also help OIE and FAO for some specific activities. And in particular, you can see here the collaborating centers for the OIE, like the training epidemiology, brucellosis, contagious bovine pleuropneumonia, and blue tongue and West Nile. And we are also reference center for the FAO in veterinary epidemiology. So these are some of our expertise. Our research is a very practical research, is mostly focused on new analytical diagnostic methods, new generation sequencing, the genotyping of emerging pathogens, because as vets, we think the link between the human disease, the animal which may be included in this chain of transmission, and then the food source are very important to be all connected. And you can all connect the same pathogen to all these three things, human health, animal health, and food only, if you genotype the bacteria or the virus, and you be sure that the source attribution is being individuated. Then we also work on surveillance and risk analysis. As I told you, many research projects are on vaccine productions and diagnostic kits production. The ministry asks us to work also on enlargement of safety and information systems and animal welfare. So this is one of the things we manage. It's important to be noted that all the animals are identified in Italy, and lately also avian species and bees are going to be registered, and we manage the database, the national database. Molecular surveillance, I was already telling you something. And we also have a national international center for training and education. So we try, above all, with some countries, the developing countries like Africa, we try to help other countries to develop the same quality standards that we use in Europe, above all when animals or food of animal origin have to be exported from these countries. COVID-19. So since the big pandemic situation last year, we converted a big lab, which is a BL3 lab of 400 square meters, which was used before the COVID situation for food safety. So in this BL3 lab, we apply technology for meat and dairy production, for challenge tests, and see shelf life tests for food productions. So from COVID situation, we needed to convert this lab in a COVID lab. So we actually run more than 3,000 analyses daily in the surveillance of SARS CoV-2 from humans, in human samples, in human swabs. So we work from Monday to Sunday on 3 separate working lines. And in this, we are one of the 3 labs, but I can say the biggest lab, helping the regions of Abruzzo and also Molise for the SARS-CoV-2 surveillance. This is an overview of our lab. So it is constituted by biobubbles, which are BL3 labs, different labs. And we needed to enlarge this lab with more PCR areas for extraction and amplification, and also with more computer for the data management. But mostly these are the 2 big biobubbles used for inactivation of samples and first part of extraction of the RNA from SARS-CoV-2. At the very beginning, when we were unprepared, we used a smaller lab, which we have. It's a BSL-4 mobile laboratory, which was constructed with the idea of use them in case of emergency. It can also be put on trains or on airplanes, and it can be moved among countries. So we started using this BSL-4 mobile laboratory for COVID surveillance. And then, of course, with the amount of analysis we are going to face, we needed to convert the previous lab, BSL-3 lab, into a COVID lab. And now we go back to the experiment I would like to speak today. So the Ministry of Health asked to the EZS in Italy to cover more research on SARS-CoV-2 during this pandemic situation. And we got an idea also for the gap, from a gap analysis, for problems that we are in having some kind of data in relation to SARS-CoV-2. So we had this idea of test the capacity of the edible live molluscs to bioaccumulate coronaviruses in general. In particular, what is known known is that SARS-CoV-2 is in water environment. We know that viruses are not being able to treat it by the classical water treatment process. So they are not retained into the depurators, but they can go to marine waters through or lake water through rivers, which these viruses can be gone from the human biological fluids. And we also know that in literature there are many data about the occurrence of SARS-CoV-2 in wastewaters. What is unknown is the ability of the molluscs, the bivalve live molluscs, to bioaccumulate viruses. Not what is unknown. What is still known is that molluscs can bioaccumulate viruses in general. So foodborne viruses in particular, enteric viruses like norovirus and hepatitis A virus are generally bioaccumulated by live molluscs. And it is also known that molluscs can be used as environmental sentinels because of this ability to concentrate some bacteria and viruses from the environment. So just to remember, live bivalve molluscs are filter-feeding animals. They are, according to the different species, able to filter liters and liters of water daily. And for this, their activity, they generally bioaccumulate metals or bacteria or viruses into their bodies. And now we pass to what is unknown. So what is unknown is the potential ability of this specific virus, the SARS-CoV-2, to survive in marine environment, because it is known about the wastewaters, but not about ability to survive in marine environments. And what is also unknown is the potential ability of molluscs to bioaccumulate coronavirus, because it is very little or nothing in literature. So we did an experiment. We performed an experiment with this design in particular. We chose like bivalve mollusk Chamelea gallina, which is one, together with Mytilus gallo-provincialis, are one of the species more commonly present on the Italian coast. So we took 150 animals from the Adriatic coast of the Abruzzo region, and we let these animals stay for 1 month in laboratory water saline conditions in order to adapt the animals to have the strongest animals that can survive in breeding situation in laboratory conditions, and in order to be sure they were depurated, so they were cleaned from any other bacterial or virus contaminants. To be honest, we went more over 1 month, so we had these animals breeded in lab for more than 1 month. Then we needed to choose a virus, which was not the SARS-CoV-2 virus, of course, because it has to be managed in BLS3 laboratory. We chose from literature virus which could be considered a good surrogate, a good spike for SARS-CoV-2. And we chose the murine hepatitis virus, which is generally recognized a good surrogate for being biologically similar to the SARS-CoV-2. So it is also coronavirus with the envelope, of course, RNA virus and whatever. So it's the proximus, the proximus neighbor to the SARS-CoV-2. In order to choose the correct amount of virus for the application in our experiment, we also studied the literature, and we decided to apply a viral titer which was giving us 1.3% to the 9 genome copies per liter, which was still into the literature data about the more common genome copies type found in naturally contaminated waters from several viruses. So we performed the growth of MHV, the murine hepatovirus, in our lab. So we were learning how to manage. We learned how to manage this virus, how to grow up on cell lines, the L929 cell lines. So we isolated them and we titered them. We reproduct this virus in order to have high quantities of the titer. In fact, there was not much problem into its replication. And then we real-time PCR protocol, specifically quantitative real-time RT protocol, in order to quantify genome copies from live virus that we were going to use in the experiment. Another important step was to find a good protocol to be sure that we were able to concentrate the virus from the water. You know, another big problem is that waters, so wastewaters or saline waters, must be contaminated also with very weak quantities of viruses, and procedure must be sure that we are able to concentrate the virus from liters and liters of water. So we tested two different methods: the precipitation with iron chloride and the use of the more expensive tangential flow filtration, the cassette for the tangential flow filtration. So we in our previous test, we started with 10 liters of water. So we were actually happy that the precipitation with iron chloride that is cheaper actually, was good enough to concentrate the virus from 10 liters of water. So we used this technique for the concentration from waters. So our experimental design, which I hope it's clear enough, it's a bit complex, but I just explain you in words. So we started from one tank containing only water. So this tank was our water negative control. Then we had another tank that contained water and mussels, and this was for checking our mussel negative control. One tank contained water and virus. This was to verify the stability and the survival of the virus along the time of the experiment. Then we had 9 tanks. 3 tanks were having virus, molluscs, and water for checking 1 hour, 0.5 time of exposure. So in triplicate. Other 3 tanks were having mussels, water, and virus in the same conditions of before, but we use these other 3 tanks to take experiment up to 6 hours. And then the other 3 tanks in parallel, which were taken up to 24 hours. So our experiment in triplicate, starting from T0, went up to T1.5, T6, and then T24 with these controls. Each tank was containing 20 mussels, in case the mussels have to be present in the tank, and the water was saline water done in laboratory, saline water in 10 liters amount. These are some pictures of the experiment. The virus is a class 2 virus, so we managed to contaminate the waters not in a biosafety cabinet, but then when we filtered the water, we also used the cabinet. This was for an excessive biosecurity attention, but it is not a dangerous virus for human. So after each tea time, we took our sample of molluscs from each tank. So our sample unit was done was constituted by the 20 muscles from each tongue. So we dissected all the 20 muscles for sample unit, and we took only the digestive glands of these muscles according to an ISO standards, the ISO 15216 of 2019, which covers the analysis for norovirus and hepatitis A virus detection from live muscles. The waters were, at the end of the tea time, were filtered, and the virus, the genome copies of the virus, were assessed, the concentration side of the water. And some water was also taken in 1 ml quantities for assess both the presence of the virus during the time and the survival of the virus during the time. And these are our very preliminary results. So they are not published yet. The experiment is just finished. So we demonstrated 2 things. And it is really a surprise because, to be sure, known the real lack of resistance of SARS CoV-2 in the environment, because it is an RNA virus and because it has an envelope which is very delicate. We didn't really think to have any result. But instead, we could demonstrate from the experiment that not only the virus was surviving up to 24 hours with a decline of 3 logs, but the muscles were also able to bioaccumulate a live virus starting from 1 hour and a half, and the maximum accumulation rate was obtained with the 5 logs at T6. Compared to other results, we took our experimental design from a paper, Italian paper, an old Italian paper. In that experiment, it was able to concentrate hepatitis A virus in Mytilus galloprovincialis in 1.5 hour time. So compared to that results in Chamelea gallina, we were able to concentrate the virus with a peak 2 hours later at 6 hour time. For what concerns the MHV survival, well, as I already told you, MHV was able to survive in this laboratory marine water up to 24 hours. And this was checked not only by the increasing genome copies with quantitative real time, but also with MHV isolation and titration. You can see on the left the conditions at which the saline water was prepared, and it can give you also an idea of what resistance parameters MHV was standing. So the temperature of 18 degrees, salinity, pH of 8.24, that were the characteristics at which MHV was able to survive for 24 hours. So what is planned next? So this experiment was going to fill a gap, at least on coronavirus ability to be accumulated by muscles and their persistence in marine environment. So should we have had positive results or not, the second step of the research was tended to monitor the SARS-CoV-2 along the Italian coast. So we are at the moment on progress to collect 250 ml samples of different species. So we go from Chamelea gallina, but also to Mytilus galloprovincialis, which is also another molluscs quite largely breeded in Italy, Ruditapes philippinarum, and Crassostrea gigas. And there are also some local autochthonous molluscs like the Cerastoderma glaucum in Sicily in particular way. The last thing, we are supposed to give results after having assessed these samples to give results for evaluating a prevalence of SARS CoV-2 of 1.1%. So without having any literature results, we planned our sampling plan in order to cover this prevalence data. So we are supposing to find 1-2 positive samples among our samples that we are going to collect. And I hope I have been clear enough and not too short in my presentation. And I thank you for the attention. And this is part of the group which has worked to this experimental. This is an experiment that took only 8 months' time from the design up to these partial results. And thank you.
Okay. Thank you very much, Giuseppe. We have some time for some questions. Looks like we've already had some come through during the presentation, but if anyone still has another question to ask, please just enter that now into the control panel and we'll try and get through some of those. So quite a range of different questions here, Giuseppe. I'll just pick the first one here. This may be going on a little bit from where you are at the minute, but do you plan to study the heat resistance of coronavirus in molluscs eventually? And do you know what percentage of molluscs are eaten raw versus cooked?
Yeah, so I want to stress an important thing that when we started this research, we were not intended to give any information on the food safety from the food safety side. So we started from the early beginning. So we wanted to know exactly if this virus could be present in marine waters, and if yes, just in genome copies or in live virus, and then if these animals could be seen as good bioindicators. In fact, our samples, I didn't tell you, and I want to stress this, the samples we are taken from the Italian coasts are all samples not intended for direct consumption. So they are taken for the monitoring quality of the waters, so for assessing, for indirectly assessing the microbiological quality of the waters. So they are just taken for this reason. Then the temperature. We are strictly faithful that the heat, of course, is a resolutive problem for any virus, foodborne virus. So the heating is something important. I also know that some mussels are not cooked for the classical 5 minutes boiling temperature, because we know that with hepatitis A virus and noroviruses is considered from the EFSA, the European Food Safety Authority, as important parameter to boil food in general, and so to cook them lively for at least 5 minutes. And it is not something that probably is going to be done for all the live mussels. So I can be suggesting for the moment to cook as the authorities advise. And for more studies on the food safety side, from the food safety side of live molluscs and SARS CoV-2, of course, if this problem will be really addressed for public authorities, I'm sure that more studies will be going to be carried out also for being sure at what temperature SARS CoV-2 is completely destroyed. Sorry, a second, I just take. So definitely it will be done in the future.
A couple of other questions more along the experiments themselves. You mentioned using a safe model in place of SARS CoV-2, which is understandable, but I think you touched on this already a little bit, but was there any other reasons why this type of mollusk was selected as the model for these studies?
We decided to choose the Chamelea gallina because at the beginning the project was only for the coast of our under our institute jurisdiction, and it is the molluscs on the sand sea more easily to be found and collected from Italian in that side. Then the ministry asked us to collect also samples from all Italy, and so we needed to change also species because you can't find the Chamelea gallina all over the Italian coast. So we chose this Chamelea gallina because of this reason, because it is mostly common on the Adriatic coast. Of course, different animal species, different molluscs filter the water in different ways. But in our experiment, we didn't want to test the trend among the species, but to see the potential of live bivalve molluscs to bioaccumulate. And the potential is potentially associable to all the bivalve molluscs.
And I think you've just answered then the second question on are there differences between accumulation of various viruses and other pathogens between the different shellfish and molluscs that are farmed commercially. So yeah, you've answered that one already. Another question. Sorry.
Yeah, just to go a bit deeper, the only standard methods which is at the European level is recognized for finding, for the detection of norovirus and hepatitis E virus from mussels, the ISO standard 15216. They just treat live bivalve molluscs like only one kind of food, so they don't specify among different species. And also when we validated and accredited the method, we used different samples from different species, but it wasn't asked us to specifically address the tests of validation on a specific species. So from my point of view, this means that does make big difference in the ability of bioaccumulating. The important is to start from at least 10 animals, from a sample of at least 10 animals, to be representative of the lot of that moment analyzed, that in that moment is analyzed. So that's why we chose 20 mussels, because sometimes 10 animals are too few for small animals like Chamelea gallina. So we had 20 to be sure that the virus could be found among the lot of production.
Another question is, do you know what percentage of the MHV detected is live and viable, or is that something that's important or not for this type of experimental research that you're doing?
Yeah, the titration is still ongoing. So the first step, we want to know if it was live. And how did we do it? So we took water samples along the times of our experimentation, and these water samples was frozen from one side, and from another side was directly put onto cell lines to see if it was replicating. And once it replicated, from the cell lines, we calculated the genome copies. And when we found that the genome copies were higher than the beginning, that was titer supposed to be at maximum 10 to the 10, but when we found 10 to the 11, 10 to the 12 genome copies, we were sure that the virus was alive, was able to multiplicate. Otherwise, we didn't have this result. So only from these samples, we went back to taking the frozen sample and to titrate the MHV. And the titration activity is still ongoing. So, but I think that it will be very difficult to give a percentage, because if you put a number of genome copies, which we know the TC50 of the original virus, then we will have many viruses will be going to die, and many others which are going to survive. And yes, probably from the titration, even resisting at frozen conditions and whatever, we can have an idea, very vague idea of the trend of survival in TC50. But it will be a result that we will have only later on.
Do the shellfish or molluscs clear virus and pathogens over time without future exposure, or is that bioaccumulation something that's longer term?
Alan, sorry, Alan, could you repeat the question?
Sure. Do molluscs clear virus and pathogens over time without future exposure to those pathogens, or is that bioaccumulation? Once they have it, does that persist for a long time? I think the question is.
Yeah. So this is another big question. You know, the food law doesn't at the moment ask authorities to look for viruses. And we know from also paper that I published that there is no correlation between bacteria and virus. So when we have mussels contaminated with bacteria, that generally we use like indicators of fecal contamination. So it was supposed that when you find E. coli or Salmonella, you know that there is a sort of fecal contamination and whatever. Well, we found that positive samples to norovirus and hepatitis A virus were negative to this bacteria. So it's not really true that there is a correlation among bacteria and virus. So first. And second, even if it's known that depuration time is okay for bacteria, it is not true for viruses, because viruses are able to constitute a very strong link inside the bivalve organs, which let any depuration system be ineffective, and it is experimentally demonstrated. So if you ask me how long you need to depurate muscle for having it clear from a virus, I should say too long for the human time, for the commercial time intended for the consumption. So there are monitoring activity in place in Europe to see if there is enough hepatitis A virus or norovirus to justify the introduction of new criteria in the food law. So for the moment, the cooking is, from my point of view, the best thing for helping us. And since I have been also in Brussels for discussions in the introduction of new criteria in the food law, I can tell you that for hepatitis A virus, we are still far from deciding the criteria. But from Europe surveillance on oysters for norovirus, Europe is going to decide for a quantitative criteria for norovirus, but of course not live virus, but only standing on genome copies. So they will be settling a limit on authorizing the commercial of some food mussels lower than some genome copies levels for norovirus, but it is still undergoing.
Okay, well, I think that's all that we have time for today. So thank you to everybody who has asked some questions. There's a few more unanswered questions, but we'll get those through to Giuseppe and he can pick those up individually later. So we really appreciate everybody joining us. Recording of this webinar will be available on our Learning Centre, on our website from next week, and you can share that with anybody who might be interested in this subject. So finally, thanks again to Giuseppe for this very interesting talk, and it was nice to get some broader perspective on the work that you and your group are involved in and how that relates to the public health system. So it's useful to see these things with some perspective, and also good luck with the rest of your research that you're doing on the topic that you've covered today. So thanks again, and thanks again to everybody for joining us today, and hopefully you can enjoy some of the other presentations on offer. So, goodbye, everybody.
Thank you, Dr. Muller, for this. for this opportunity. Thank you. And my mail is at the end of the presentation for anybody who wants to have more information or test for collaboration. So we are free to do anything for the public health. Thank you.
Okay, thank you. Goodbye.